So in previous video we have discussed about RFLP marker, that is restriction fragment length polymorphism, and in the end of that video I have told you that the major disadvantage of RFLP is that in some situation when you are not having too much amount of sample, you cannot perform RFLP. Okay, and there are at that time no option to amplify it, because I have told you that RFLP is a marker which is developed in pre-PCR area. Sorry, RFLP is a marker which was developed in pre-PCR era. Okay, so under such circumstances we have no any instrument to amplify it. So today we will going to discuss about AFLP, which is nothing but a modified version of RFLP. Okay, that means when you will combine RFLP with PCR technique, you will end up with AFLP. Okay, so I will also upload another molecular marker video very soon, that is RAPD. So RAPD is the molecular marker technique where we have first used the PCR. Okay, so we should say that AFLP is the combination of RFLP and RAPD. Okay, how? Because in the very first video we have discussed that RFLP is an example of dominant marker. So what was dominant marker? Dominant marker is the marker technique who have the ability to distinguish between dominant and recessive, right? And not only that, it can also help you to distinguish between what is polymorphic DNA and what is monomorphic DNA. We have discussed about polymorphic DNA and monomorphic DNA, right? So polymorphic DNA means suppose it is an individual X and this one is individual Y. Okay, so under such circumstances polymorphic DNA should differ in case of X and Y also. Okay, that means it is a particular region of DNA that is present in both the X and Y but differs among them, right? We have talked about VNTR, we have talked about STR are the example of those kind of polymorphic DNA. That is, VNTR is present in each and every person in this universe. VNTR is present in me, VNTR is present in you, VNTR is present in your friend and everything, right? So, but the length and the repetition or the copy number of the VNTR varies in each of the individual. Okay, and that is responsible for the 0.1% variation which is present in each and every individual in this universe. Okay, so that is the idea, that RFLP is a dominant marker. First thing, it will help you to distinguish between dominant and recessive and also the polymorphic and monomorphic gene. Now the question is, what is monomorphic? So monomorphic DNA means similar in the case that we have discussed in polymorphic DNA. So again let's consider a individual X and Y. So monomorphic DNA is the particular DNA present in particular chromosomal region, okay, which is similar in every individual, okay, and give you a similar band pattern when you will run them in agarose gel electrophoresis. Okay, so that means the exact opposite of the polymorphic DNA is monomorphic DNA. Okay, and so we have learned that RFLP can distinguish polymorphic DNA. Okay, and now it is important to talk about RAPD. So for now you have to only remember that in RAPD we are using PCR. Okay, but another important thing that is important to remember, that RAPD is a non-specific PCR technique. Okay, that means the primer that we are using is not specific for a particular sequence. Okay, because in PCR videos we have discussed that PCR is responsible for the selective amplification of a particular part of the DNA, right? But in RAPD we are using or we are performing non-specific PCR. Okay, so now we are combining RFLP and RAPD together, right? So RFLP have two steps we have discussed in earlier video, that is, one is restriction endonuclease digestion, and another one is agarose-gel electrophoresis, by which we are visualizing the length of the polymorphic DNA or the variation in the length of the polymorphic DNA, right? And in RAPD we are using PCR. So if we combine these three steps, then we will end up with AFLP. Okay, so another important thing that is necessary to understand, that as we are using restriction endonuclease, and we have already discussed in enzyme in recombinant DNA technology video that restriction endonuclease can cut DNA specifically, right? Restriction endonuclease is a specific endonuclease, right? So the end that are generated is already known to us, right? So here the PCR that we are using is obviously a selective PCR. Okay, that means here the primer will allow the amplification of a particular part of the DNA. Okay, not non-specific amplification we will observe. So now, so that is the idea of amplified fragment length polymorphism. Okay, that means here we are visualizing the polymorphism of a amplified fragment. That is the concept of AFLP. Okay, and that is the combination of RFLP and RAPD. Again, remember, RFLP is a dominant marker and it have the ability to distinguish between dominant and recessive gene, and it have also the ability to distinguish between your polymorphic DNA and monomorphic DNA, but it cannot talk about anything about the allelic variation. Okay, so in RAPD we are using PCR. Okay, I hope you can remember that dominant and recessive allele distinguish is done by dominant marker. We have seen in very first video that in a parental testing we have get that the father's allele is dominant and mother's allele is recessive, and we are getting the child's band pattern either similar to father's band pattern or similar to mother band pattern, right? And from that we can distinguish about only that whether the allele that the children contains is the dominant allele or recessive allele. Okay, but we cannot tell about the genotype of the children, right? And that problem was solved later by codominant markers, right? For example, SSR we will going to discuss. So now it is important to understand about the various steps of the AFLP. So first, this is the genomic DNA. So before that, there should be another process, that is, this is the cell. Okay, so we are isolating the genomic DNA. After genomic DNA isolation, it is important to apply restriction endonuclease digestion with at least one or two restriction enzyme. Okay, so which will generate fragments, restriction endonuclease fragments. Okay, now as you can see, there are staggered end, right? And it is obvious because most of the restriction endonuclease that we are using creates staggered end, right? Because it is easy in ligation purpose. Okay, so now as I have told you, we will going to apply a specific PCR. So now it is important to understand about the sequence, right? So now we will apply a trick that we will use adapter. Okay, so adapters are nothing but short DNA sequence and which is ready-made staggered end. Okay, so now let's try to understand if we use those ready-made staggered end and we will join them. So what we will get? We will get a blunt-ended DNA fragment, isn't it? Okay, so now if we zoom those blunt-ended DNA fragments, so now we can easily understand that in both the end of each of the fragment there are our adapters, right? So now as we know that what kind of adapters we have used, so it is very easy to construct a primer on the basis of the adapter that we have used, right? So this is our forward primer and this is our reverse primer. That is the two important primer of PCR. We have discussed in PCR video. If you don't know about what is PCR, then I would like to recommend you to go and watch the PCR video lectures, then it will be clear to you. Okay, then we will perform PCR techniques and PCR will perform amplification, right? And that's how we will get amplified fragments. Okay, so after getting the amplified fragments, the step will be we will run them in a agarose gel electrophoresis. Okay, so we will get DNA fragments. Okay, now from that DNA fragments we will perform Southern blot. Okay, Southern blot means here we are transferring the DNA fragment from gel into membrane. That can be nitrocellulose membrane, that can be nylon membrane. And it is important to understand that we are doing this because further manipulation in gel is very difficult because gel are fragile. Okay, so under such circumstances we are transferring our DNA fragment into a membrane for easy handling. Okay, so after transferring into the membrane, now we can denature them with the help of alkali. Also we can denature them during when they are in gel too. Okay, before the transfer process we can do both things, right? So we can apply alkali treatment for the denaturation. Okay, so in both the situation we will end up with membrane containing single stranded DNA. Okay, so now it is necessary to construct the probe, which probe contains P32 radio labeled at its 5' end. Okay, now when we will add those probe, this probe will hybridize with the single stranded DNA already present in the membrane. Okay, and as a result, when we will put a X-ray film on the membrane, we will get autoradiograms. Okay, and now by looking on the band pattern that we have obtained in the autoradiogram, it is very easy to understand about the DNA sequence variation in between different individuals, right? And that is the idea of AFLP, right? That means the idea is simple. It is nothing but a Southern blot technique itself, right? But here the extra addition is the PCR. Okay, for just increasing the amount of the fragment for easy understanding. So let me told you that with the help of AFLP technique we can get 50 to 100 bands. Okay, and those bands are easy to analyze as compared to RFLP and RAPD. Okay, and also AFLP can produce a highly reproducible and accurate result as compared to RFLP and RAPD. And important thing, no prior information about the DNA sequence is required because we are using adapters, right? And these adapters will solve the problem of using primer, right? Because we can construct primer on the basis of the sequence of the adapter. Okay, so important thing that with the help of AFLP we are getting near about 50 to 100 bands. Okay, and now by comparing those 50 to 100 bands, it is easy to understand about the variation in DNA fragment between different individuals. Okay, and it is relatively easier to analyze as compared to RFLP and RAPD. And AFLP produce a highly reproducible and accurate result as compared to our RFLP and RAPD, and no prior information about the DNA sequence is required. So in short, if we want to revise this, then AFLP is a amplified fragment length polymorphism. Okay, so AFLP is nothing but a mixture of RFLP and RAPD. RFLP was the dominant marker that we have discussed in previous video, and this RFLP contains a major problem that if we don't get large amount of DNA fragment, then analysis becomes difficult. And as it is a discovery of pre-PCR era, so we have not any amplification process, right? So as a result, we have to do this with that small amount of DNA fragment, DNA samples only. But after the development of the PCR, we have mixed RFLP with RAPD. So RAPD is the first molecular marker technique developed using PCR. So now this PCR technique from the RAPD is adopted, and from the RFLP we have adopted the restriction endonuclease digestion, and then by combining these two steps we have constructed the AFLP. Okay, so this AFLP has a similar work to done as we have discussed in RFLP, that it will differentiate between polymorphic DNA and monomorphic DNA. Not only that, it will also help you to differentiate between dominant allele and recessive allele, but it cannot show you any allelic variation. That means whether a person is homozygous or heterozygous. Okay, and that was the major disadvantage we have discussed about the dominant marker, right? So after that we have learnt about the process of AFLP. That first step is the cell isolation. From that, with the second step is the genomic DNA isolation. From that genomic DNA isolation, it is important to apply restriction endonuclease digestion with approximately 1 or 2 restriction endonucleases, okay, that will create fragment. Now we can run apply PCR. Okay, before doing PCR, it is important to add adapters. Okay, these adapters will help us to use specific primer for the PCR technique. Okay, and then PCR we will get amplified product, and we will run those amplified product in agarose gel. Okay, and we will get bands. Okay, so now we will apply alkali treatment, that is NaOH. This alkali will denature the DNA fragments. So now in the agarose gel we have single stranded DNAs. Okay, clear. Now from that we will apply southern blot technique, and we will transfer them in nitrocellulose membrane. And okay, and in nitrocellulose membrane now we have single stranded DNA. Now we will construct the probe, which is radioactively labeled at its 5' end. Okay, and now with the help of this probe and autoradiogram, we will get a autoradiograph. This autoradiograph will contain DNA band patterns. Okay, and now with the help of this DNA band patterns, we can analyze the variation between different individuals. Okay, so that is the idea of AFLP. So thank you for listening to this class. We will again come back with another molecular marker, that is RAPD.